Hello, I'm Frank Helle, and I will show you how to write a practical called the Osmosis Practical as if it was an IA. And the purpose of this is to let you train how to make the IA in a good way, but at the same time meet all these criteria, such as personal engagement, exploration, analysis, and a little bit evaluation, and also how to write it in an understandable way. So in total you can get 24 points if you write your IA perfectly. One advantage of starting with the practical as a kind of a practice exercise or practice IA is that you can practice without the stress of managing experimental design yourself, because that can be hard. Also, if you are in my class or if your teacher provide this box, you have these sets of tools that can help you write this first mock AI. This is the title of the mock AI, and it's not a question. So the title is not the research question, which we will come to later. In the introduction, you start wide, and then you narrow in on what is relevant for you. So for example, here you could talk about osmosis. But for example, when you come further down, you could talk about the plasma membranes of potato cells in particular. Also, it can be a good idea to have a figure in the introduction showing kind of the main phenomenon you're exploring. And a good figure has annotation, and also it has a name. And in an IA, it's best to have just only two different names for elements in your study. Either it is a table, if it's not a table, then it's a figure. So if you have a tracing, or if you have a micrograph or something, just call everything a figure unless it's the table. And then we see here we have the description. Another thing, do you notice that on the figure everything is written, both the name and the description is on the bottom? That's a convention. And also tables, they have the opposite placement of the name and the description on the top. So here then we have the aim of the study. What are we intending to do here? Then the research question, and I will come back to a very good way of designing the research question. But then we come to the hypothesis, and the hypothesis is that you say what you think will happen based on your knowledge in the subject. So in the potato experiment, we will have 3 different saline solutions, and I say in all 3 situations what I expect to happen. And we also need a null hypothesis, meaning if we don't find the results we are expecting, what does that mean? Now we come to the variables tables, and notice again, since this is the first table, it also has a name. And remember the counter, it's chronological. So since this table 1 is the first table referenced in the main text body, the main text body is kind of this text here, it is number 1. Also see that this table is named on top, and look also that it has a description as normal. And then the next table referenced in the main text body would be table 2, and so on. And in the variables tables we have the independent table, and that's the thing we change. And in this case we change the salinity of the solution where we drop the potato cubes. Then the dependent variable is what we measure, and we measure actually change in weight. And to do that we have to weigh the potatoes 2 times, one before and one after incubation. And then But what does this mean, this change in weight? Well, from it we can deduce the direction of osmosis, and that's a derived variable. And the derived variable is kind of the thing we conclude about in the end. So for example, another situation, let's say you measure heart rate and ventilation rate, that is breaths per second, after running stairs, and you're testing teachers versus students and their how they react on running the stairs. Then the dependent variable would be ventilation rate, heart rate, of course, but the derived variable would perhaps be stamina. And then control variables, I think we try to keep constant in the experiment. I told you we would go back to the research question. A really good recipe to write a research question is, how does, and then you just insert independent variable, the one we change, affect, and then we can put in the dependent variable as a measure of, and then you can maybe put in the derived variable. So if I did this directly, it would be, how does saline concentration, parenthesis 0, 20%, affect relative weight change as a measure of the direction of osmosis, for example. That would be a great RQ. And do you see how simple it was? Just by making the variable table first and then the RQ. Then let's go up here and look what I finally arrived to. So you see I've switched up things a little bit. You can do that after, but you saw that I had a really good starting point. So my RQ now is, how will osmosis in different solute concentrations affect the relative weight of submerged potato cubes from solanum to bursum? And we see here, instead of just saying potato, I use the Latin name. And when I do that, I use italics and capital genus and not capital first letter on a species. Also, I use units. This is a special case since I use a relative measure, then the unit cancels, but I include them anyways to show the examiner that was not by chance that I left out the unit when I could. And also I try to define everything as well as I could. Okay, so here the potato cubes was about 0.13 cubic squares, so I put that in. I could also take in an approximate weight or something. And also I'm specific, so I don't say just different solute concentrations, I say the specific concentrations of those solutions. But still, having all this detail, it's pretty short and concise. Actually, then we come to the next section, which is methods. And in the method, you can include a flowchart. So here we show the potato experiment that we cut 8 cubes, weighed them, incubated them, weighed them again, and so on. And it's not compulsory to do it in your IA, but you can often get a lot of goodwill and maybe an extra mark for doing it from the examiner because they love seeing this stuff. Then the materials list, then you list the things. But the thing is, you have to provide accuracy of the weighing equipment. For example, the electronic scale had 3 digits, and that means that this final digit will kind of be rounded up or down depending on this hypothetical digit here. So if this is kind of more than 0.5, this will round up and so on. So that gives a rounding error of 0.0005. Then this figure is actually obligatory, and this is the picture of the experimental setup, and it is annotated. So what does annotation mean? It means that you have an arrow or something, and then you have an explanation, and it shows kind of a relevant picture of what's going on. For example, here are the forceps that we use to move the potato bits with without getting salt from the hand and stuff. Here is the Pasteur pipette that we filled the multivalves with, and so on. And the multivalves, of course, where we had different wells with different concentrations of saline, and made us able to put each cube in a controlled environment and then have it numerated so we could compare the weights before and after. So then you come to the step-by-step method, and in contrast to the list of equipment, here it's numerated, because these steps only make sense if they are in the in a specific order. And this list should be so detailed that another student in another school which don't know you could repeat this experiment. In the safety considerations, you should write about the safety for you if you work on conscious animals or insects or something, like the ethical use of they, them, also the safety for the environment, and then also ethics. And ethics is things like data security, like if you have biometric data about students, like their bacteria flora or something, you should not reveal that. Now we come to the results. And here, first, we have a table with all the raw data. So, for example, here we have kind of the cube 1 measured the first time, and then we calculate relative change. So, for example, absolute change would be, if this is M1 and this is M2. Absolute change would be M2 minus M1, simple. But since the cube varied in size, the data would vary less if we used relative change instead. And relative change has this formula. It's M2 minus M1, and then you divide by M1 again. And I could have maybe made a table with both raw data and the processed finished data, but in this experiment I chose to have the table 2 and 3 separate, because in table 2 you can write about that the individual weights of the cubes varied, and so I just reference table 1 when I talk about the raw data, outliers, stuff like this. But then when I talk about the conclusion of the experiment, what we found, the kind of the over view of the data, then I have that in table 3, so I can much more easily reference the data in my text when I divide it up this way. So we see that in table 3, yeah, so this should be only salinity here, and we shouldn't have this, that's just confusing. So these are the salinities, the fluids we put the cubes in, and then here on the first row we have average relative weight change. Yes, it's this value, plus minus standard error mean, and I will come back to that later, but that means the standard error mean talks about the spread in data. So for example, if I measure something and I measure these values here, that is a large SEM, and for example, if it was expressed like an error bar, it would be something like this. But if we have the data very closely together, it would be a small SEM and the error bar would be small. In this video, we are going to use calculate standard deviation instead, because that's more often used, which is same type of thing as SEM, but standard deviation is more about where you can expect a single value to be in the dataset, while SEM is more where you can expect the average to be in the dataset. Then the next row, we see that we have number of potato cubes. So remember the flowchart, that there were 8 cubes in each experiment, or we can see it here also. 1, 2, 3, 4, 5, and then 8. That's a good thing to have in the main data table, because number of measurements is very important for the statistics. The more measurements, the better statistics. So for many examinators, they look often more on this than, for example, the values. And then we come to the statistics itself. So here we could have used a t-test if there were only 2 groups, but since there are 3 groups, we can't use a t-test. We have to use an Anova, which is kind of a t-test expanded for more than 2. And this p less than 0.01 means it's significant. And you might wonder, what does that mean? Well, It means if you repeat the experiment, you are likely to find the same result. To be specific, if p is less than 0.01, it means if you do this experiment 100 times, you will get the same result, that the increase in 0% is larger than 1 and 20, 99 out of 100 times. So that is pretty good, solid data. And here, this only shows that the drop in weight in the 20% saline, the hypotonic solution, is significant both if you compare it to 1% and if you compare it to 0%. Now we come to the main data figure, and since it's a figure, the description is on bottom. We have the name and a short description. Also we have the group names here. Here is the unit, relative weight change, maybe that should have been on the left side. And we see that we have when the weight drops, so when M2 minus M1 is less than 0, of course divided by M1 since I use relative weight, it's a negative change. It's really important that your main graph reflect the direction of the change. We also see that this is almost 0. And that is very interesting because the 1% saline is supposed to be hypotonic, and in hypotonic solution there should be no change. So it would be interesting to see if that is significantly different from 0. And now I'm actually going to show you how to make this graph, not in Google spreadsheets, which this is made, but in Excel. So remember these data. Now I have these data here. So this was the 0% start end of the potato cubes, and here are the weights. So now we are going to calculate relative weight. So that we start in Excel by writing the equal sign that signals that now I want to make a formula. I make a parenthesis and then I take M2 and weight minus M1, the starting weight, and then I divide it by M1 again, and now I get relative weight. If I was not so concerned about using relative weight to reduce spread, then I would just take M2 minus M1 and skip this whole divided by M1 thing. And when I have one cell, you can just drag this formula along the whole row to get all the data. And then I can copy this to make to transpose paste. That means I switches columns and rows to get data here. But there was a problem because here are formulas, so I have to first just paste values, copy again, and now I can finally transpose paste. So here I have 0%, 1%, and 20%. And now I want to make a graph first, then do the statistics. So what I will do here is that I will calculate the average. Again, I could have added all the 8 values and divided by 8, but instead I'm going to use the built-in formula. And I select the 8 values and I get the average. By double clicking I can confirm that it's the correct range. And now I'm going, so this was average. Then now we're going to calculate standard deviation. We start with equal sign and we use the unsupported standard dev formula, and we see that there is a pretty large standard deviation here. And now we will copy these and get the data for both groups. To make the graph, we go in the insert menu, and then we select the simple graph. And if we don't have any data selected, you should get an empty graph like this. We right click and choose select data. And sometimes you can just use this one to drag series names and values, but since we have percent, it will interpret this as data as well, so that won't work this time. So instead we have to do it the hard way. So we started with this one, we press add, and then we specify that the series names are these ones, and then that the values are these ones. And now we should get the correct graph, which we did. So here we see 0% and 1% and 20%. Now we can try to change this. It was not a very useful title. So we could say relative weight change. And we see that we need axis titles. So we select this. When you click on the side of the graph, you get this menu here, which is actually where you can add stuff. Many things you can add it here in the sidebar, but no, that's only for formatting things you have already added. So, and the first thing is the axis title, which is a relative weight change again. Gram divided by gram, since it's M, the difference between M2 and M1 divided by M1. And then here we have saline solution. Concentration maybe. And then we see that since we have a negative bar here, the grip names are a bit confusing. So we will select the thing we want to change and then go on the format button. And since these are labels, they say this is something, this is the name, this is named 0% and so on. We go on labels, and then we go on label position, and we will have them low. And then we have this nice standard variation. So how can we get those into the graph? And that is the reason why we're using Excel and not Google Spreadsheets, because this is much easier to do in Excel. So first we had to add error bars. So they are here, plus sign, error bars. But we want to use a specific type, not whatever this is. You go on more options, and if this is on the error, like the rightmost pane or tab here, you will get this directly. Otherwise you have to press this one. And we want custom error bar, and we have to specify. We say this and this. Then we press OK. So now we see that the error bars actually reflect the standard deviation. So this is kind of 0.03, which is almost 50% of this one, which is correct. The one losing water in the hypotonic solution, you see there is much less spread in the data. We also see that 1% has an error bar that crosses 0. So this is likely not significantly different from 0. Remember that the data table showed that 1% and 20% were significantly different. So I'm going to add a significance indicator, and basically it's just a text box. So I go on the insert menu and I press text box, and then I just make a text box with a star in it. And then I put 1 star on the 1%, and then let's see, now this is very hard. And then finally I moved the second star to the 20%, and I press Shift S Windows key on the keyboard to get this very convenient screenshot tool. And now I go back to the famous potato experiment, and we go down to the data figure, which we will now update with an Excel version. So notice that now we have changed from standard error mean to standard deviation. So we have to indicate that with standard deviation in potato cubes, blah blah blah. Now we are going to actually do the statistics that we did here to indicate that these two values were significantly different. And so we go back to the data, and these were the 0% values, the increase in 0% or distilled water. And now we will go to a web page called Vasostats, where you can do an Anova, which is the version of the t-test that supports more than 2 groups. So we have 3 groups here, so we write that in this box. We say that the samples are independent, meaning it was different cubes in the 3 solutions. We use a weighted analysis. You always choose weighted. And then we paste the first set of values. We go back to Excel, and then we take the 1%. I remove the empty row, and then 20%. And now we press calculate. And this might look like a wall of text for you, these tables are very abstract, but I will take you through everything. So this column here is just the means, and they are the same that we have already calculated. And then we recognize the standard deviation, which is this one, and even this one, the standard error, which was the one we started with. And these are things that you will recognize after you have done the t-test exercise in the statistics chapter in person. You will know what the f-value is and stuff like this. What we will look at here is this p-value. This p-value just says that there is a statistic difference in the data set, meaning the lowest value is significantly different from the largest. And in this case, the 0% had a plus 0.072, while the lowest had a decrease of minus 0.015. So at least we know that 0% is different from 20%, but are, for example, any of the groups different from the 1%, and that is what we see in this box, which is the most important. This is something called the Postdoc Test, and it measures if there are significant differences between the individual groups in the dataset. And look here, they have renamed the groups for us, so 0% is M1 now, M2 is 1%, and so on. And those two are different from each other because it says P less than 0.01. For some reason, students that I have taught to use Vasostats, they suddenly rename their groups to M1 and M2. Don't do that. Keep your naming consistent and stick with 0% and 1%. Here we see that 0% and 20% is different. We knew that from this value. And then that 1% and 20% are also different. This is why we can put these stars on both. And now we have to explain what they mean. So this star means P less than 0.01. Do you agree that was what we just saw in the statistical report? Versus all. In star indicates that this value at 1% is significantly different from 0% and 20%, which are all the groups. And this is what we already wrote here, so that's okay. And now we come to the statistics chapter. And first we say what test we used, and with a reference, that's good. And then what tool we used, because we didn't do these calculations by hand, so we have to reference this web page and the name, the guy who made it. And then remember we clicked on independent samples and weighted. Those are options that we have to specify. But then remember we are interested in if this value is statistically different from 0. To find out if that is the case, we have to go back to Anova and see if there is a statistical test that can help us determine if an average is significantly different or not from a specific value. And we have to, but then we will use this single sample t-test. And here we can enter the 1% value and then the hypothesized mean of 0. So we go back to our values, and this is the 1%. We go to the single sample t-test value. We paste the data. We put in 0 here and we press calculate. And we see that the p-value for the two-tailed test, which is the one we are always supposed to use. If you want to know more about that, look up my statistics video on YouTube called 22 Statistical Tests for the Extended Essay and the IA. And we see that it's larger than 0.02, meaning it's not significant. This means basically that if you did this experiment, you can assume that this value actually is 0, or 0 is within the range of this average. So that's what we're talking about, to test if the relative weight increase in the 1% was significantly different for 0. We used blah blah blah. T-test also from Wasserstats. And then lastly we say that the alpha was 0.05, meaning that as long as we can expect to see the same results 95 out of 100 times we do the experiment, we say it's significant, and this is the normal range or limit in biology for what is considered significant. Then here we have example calculations. So every calculation you do in your IA, you have to first show with the formula and then show with the example calculation. So we see that this 0.071 value is the one we get here, and we use these two numbers. And then even average formula has to be shown, and then here I've been a little bit lazy, so I haven't shown how you calculate standard deviation. Now we come to the discussion, and in the discussion you just go straight to kind of maybe just explain here that what the solutions were, that they were hypotonic, isotonic and hypertonic and so on. And then I explain a little bit about the cell wall, that it's flexible, so if water goes in, the whole cube might bulge out and gain weight. And then I just go directly on answering the RQ. So I state the RQ and then I just answer it. Nice and simple. And then I don't write so much more. Here I just have a justification why I used relative weight instead of absolute weight change. Then limitations, very important part of the study. And instead of writing about how I could have been inaccurate while making the saline solutions and so on, I understand that this would affect the real data very little. So instead I talk about the residual fluid on the cubes when you remove the cube from the bathing solution. That could be a significant fraction of the weight of the cube. Also I try to use my biology, so I try to talk about how cut potato cells could actually be damaged by the cutting and leak solutes into the solution and stuff like that. Then I talk about improvements. Yeah. So we see here that I'm actually lacking a chapter, so I'm lacking the strength. So you should also talk about the strength of your study. So here I would say that so strength 1 using relative reduce the, and then I would write this strength and maybe second strength and so on. Then you have to write the improvements paragraph where you tell how could you perform this experiment even more accurately or more efficiently or with less caveats. Then we come to the bibliography, and whenever we mention the biological fact or something, we have to use in-text reference, and I will show you how this is done. So let's say I want to put a reference here. Then I go on my Zotero, which I have already installed. But since I might not have the reference I'm looking for here, we will find it first. So your librarian might help you install Zotero. I'm not going to show you how today, but this is the Zotero program. So here I have all my references, but I'm actually not going to use this interface to add citations, which means the sources, the papers or whatever books I'm going to cite. I'm going to use that with a small icon here, which is called Save to Zotero. So now I'm going to go on a biology site called JSTOR, but you can probably use a lot of stuff, whatever your school has access to. And now, if you noticed, we have a folder here. So we're not ready to save a citation first. We have to enter a specific journal article. And now we got this page here, Save to Zotero, and you just click it once and it's saved. Like and if we now look in Zotero, we see that we have this citation ready. Actually, it was there before, and now we have two of it. And now I'm going to try to insert it into my document. We are using Google Docs, and now I'm going to tell Zotero to add this citation. On another screen I got this pop up, so I have to log in with my school account. And now it looks like the page froze, but actually it's just waiting down here, because now pop-up hid behind the Zotero icon and I have to activate it. And this is pretty poor user interface, because now I just have to write something that is that paper. Like, remember the author, you might notice his name was Krause, so I can write his author name, or just potato, and then one potato, two potato, whichever way I find it. If it's by year or journal or whatever, you have to choose it, and then you have to know that you are supposed to press enter here. And then this happens, and this will only take a short while. And now we see that we have since we're using the Chicago style, we have the text down here, but we have also the reference here. And if we go to the very end of the study, we can also see that it is in the final bibliography. You have to add this bibliography the first time in a document, and you do that by Zotero add edit bibliography. Then we come to the appendix. And personally, I would have pasted the output from the Vassastats page here. Instead, I've just shown you kind of a quick guide how to use Vassastats. And also in the appendix, it's wise to have stuff like the consent forms if you're using experiments on people, or lists of ingredients of things you are using, stuff like that. Everything that is kind of support for your study, but not the results itself. So now I want to go back and just show you the resources. So remember the box with the references I showed you? So you can use these checklists to go through the mock IA and check if everything is okay. So you can either choose this easy checklist or the exhaustive checklist. And the way you use it is that you look at the keyword here, and then you look up, for example, the criteria for the IA or instructions you have been giving, and try to determine, do you have a personal link in the introduction, and do I do in-text references and so on? Have I labeled the images? If I didn't, you press missing here, or needs work, you say it here. For example, when it comes to personal link, I have written this paragraph here about blueberries, which is kind of my link to osmosis. So I hope to score the points through this paragraph. So you might wonder, what have I based this video on? And I would say two things. One thing that is that I've been a researcher for 10 years in the field of renal physiology, where I worked with high blood pressure and how signaling between different tissues in the kidney, especially in blood vessels, can affect that. And I've wrote, for example, a lot for American Journal of Physiology. So I have kind of taken with me some of the conventions from there, but mainly this video is inspired by this file, which is the syllabus guide for biology for this session. And if we go to page, let us see here, page 148, you can read about what is the point of the IA, and then if you scroll a little bit down, you will come to how it's assessed. So that starts at page 154. And this will be very useful for you to go through because this is the same source as your examinators and your supervisors use. I will try to make available all the links to the resources I used in this video, such as the checklist, but my Dropbox won't allow me to share file with thousands of persons, and I've been locked out from using web links on Dropbox before, but I will try to find a way to share the link. So if you really want access, for example, to the exhaustive checklist or something like that, write me in the message section and perhaps suggest a suitable way to share the files with many people for free, and I will consider doing it. For now, I will put the link to the document I scrolled through, the YouTube video I made, and maybe some other resources. So when you do your real IA, so this was just a mock IA from a practical, which is obligatory in the biology IA course, but when you write your final IA, the most important thing for you to remember is that everything you do is has the purpose of answering the research question. Other than that, all those arguments should be clear and logical and concise, and not too verbose, short and concise. If you remember that and maybe learn the techniques that I showed in this video, I think we'll do very well. Good luck.